Hi everyone. In this video, we will explore how to identify marker genes of single cell clusters. So in the past video, we have seen that we took the single cell dataset and we clustered it, and then we visualized it using PCA or TSNE and UMAP. We talk about which method when to use. So now once we got the cluster, the next step is compare each cluster with the next cluster and try to find important marker genes. So that's what we will be talking about, and this is a very important step, and the final step we would say to identify important genes, and then the next step would be to annotate those marker genes and identify the cells. So in this video we will primarily focus on the marker gene identification. In the next video we will talk about how to annotate them. Okay, so these are the steps that we have followed. So we took the typical workflow, like you take a raw data, you do the filtering, normalization, scaling, identify the top variable genes. We do the PCA, and that will compress the data. Use PCA to do the clustering and visualize your cluster using either UMAP or TSNE, but you may go with the UMAP visualization. But once you see that, okay, so in your data you have, let's say, 5 clusters, 10 clusters, then it's very important to compare each cluster with the other clusters and do the differential gene expression. So in bulk RNA sequencing, right, so we have like the differential gene expression is very different, where we have, let's say, control versus treatment, so like that we do. And here we are doing the same thing, but here we are doing with cluster 1 versus cluster 2, or cluster 1 versus cluster 3, like that. But now you can imagine that we have in the previous example, in this PBMC dataset, we got around 10 clusters. So now if you do compare one versus other, so it will be 10 into 10 minus 1 by 2, almost 45 or 50 such comparisons will happen, and that many differential gene expression will happen. So that's the reason we don't do usually one versus every one cluster with all the other clusters. So instead what we do, we take first cluster and then that cluster versus rest all cluster as one group. So basically we let's say you're taking cluster number 1, that means the other group will be 2, 3, 4, 5, 6, 7, 8, 9, 10. So like that. So now this group versus rest all, you're doing the comparison, and whatever the differential gene you find, so that genes are the marker genes that differentiate these clusters from the rest all of them. Now same, you repeat the second process second time. So take the second clusters and compare with all. Take the third one, compare with all, like that. So that is one way. The first thing that we should do, like take each cluster versus rest. So that will give us some crucial identification marker for each clusters. But then you look into your UMAP plot, and if you do not able to find out let's say these two clusters are very close, okay, in the UMAP plot, so then you can do cluster 1 versus cluster 3 alone also you can do. So that way, one versus all is one approach to do, or alternately you can do pick up 2 clusters and you can do the clustering differential analysis using that as well. Okay, so first question asks, like, what are marker genes basically? So marker genes are nothing but these genes that are highly expressed in one cluster compared to the other, or the other way around. Like let's say you are focusing on cluster number 1, here the gene is up, but rest all that gene was not expressed, or other way around, in all other clusters the gene was high, but here your gene expression is low. So that also, both positive markers and negative markers you can get it. Okay, but usually we are interested towards the positive marker because the gene is expressed in that particular cluster, but not in other cell. So that means because of that expression of that gene, that particular cell is we are identifying that cell. So usually we take the positive, but you should also sometimes look into the negative markers as well. But marker genes are that genes that are identify that particular cluster. So marker genes help us to determine the biological identity of each cell clusters. Okay, so now that's why to do that we do the differential gene expression. So in DGE compares, like gene expression in cluster A versus gene expression in all other clusters. So the main goal is like find the genes that are highly expressed in one cluster, whereas it is low in the other clusters. And there are 2 functions in Seurat. One is FindMarkers and FindAllMarkers. FindMarker will compare between, so here you can specify cluster 1 and cluster 5. I want to compare these 2. So here you can specify that. Whereas FindAllMarkers will do like 1 versus all. 2 versus all, 3 versus all, 4 versus all, like that. So that means it will do all clustering and it will give us all the markers of all the important markers of all the clusters. So usually we don't do this one. Usually you go with this one. It will give you all the markers, and then if you are very much interested in comparing 1 or 2 clusters, then you go with this find markers function. Okay. So now, yep. So basically, what statistical test is usually used? Default, Wilcoxon rank based, test. This is a non-parametric test. That means we don't use mean median based comparison. We compare based on the rank. So that's why it is called non-parametric statistical test. And yeah, so that is much better than other. But alternately, we have other tests as well, like mask test are there, DGE test are there. So that I'll show you what all are available. And as I said, we have this parameter called a function called FindAllMarkers. So here you pass your data, and there are some other filtering criteria are there, like so you want to say that, hey, you do the differential expression but only give me the positive marker. That means whichever genes is overexpressed in my cluster, whereas rest all, right, the other group there is less expressed, so only give those kind of thing. So that's what you specify, only positive equal to true. Another parameter called minimum percentage. So basically when you are doing differential gene expression analysis, right, so no need to consider all the genes, right? So that means you can easily filter out, like a gene must be expressed in let's say 35 or 34 percentage of cells, then only consider the genes for the DGE analysis, otherwise remove them. So those kind of filter you can tune over here. So the default could be 0.25. And the third is log fold change threshold. So that means you're saying that when I'm comparing this group versus this group, and I got expression, let's say this one, I got expression of this cluster, this one, how much fold change, log value of that, if it is greater than that, then only consider, otherwise ignore that gene. So those kind of we filtering we do. So genes with high fold change between this group versus the high fold change and very low p value, that means more statistically confidence level is there, those are the strong marker candidate. Okay, so that's the thing we do. And once we identify that marker, then the next step is to visualize them. That so there are different visualization are there, like dot plot, and we can have the reads plot, we can have the feature plot, we can have the Violin plot, heat map, so that we will see where we can see how the expression of the genes look like. Okay, so to start with, this is what the tutorial says, like we have the find all marker function as well as find marker function. So if we go with find marker, okay, so here you can specify cells dot one means like which cluster versus which cluster you are comparing. Okay, so yeah, so cells dot one means like just checking, I think that's the no, that is the, I think, different, my bad, that I think identity dot one, identity dot, I have to look at the code, my code. Yeah, I think so, that's the name, I think the cell 1 cell 2 are the name probably. uh, yeah, vector of cell names belonging to group 1 and vector of the cell name, the barcode name, right? So those are, so we don't use these two. So here you can specify which group to which group compare. Okay, identity 1, group identity, and cluster identity 2. Okay, so that is the thing. And here, as I said, we can use you can change what method you want. By default is the Wilcoxon. We can have Wilcoxon lemma based method, or ROC is classification based classification based differential expression marker. It will say you can do t-test, negative binomial, Poisson distribution, masked, it's more for single cell RNA data actually. And also you can use the DSeq based differential expression as well. But the default one, Wilcoxon will work well, because that is more of rank based do that. And recently in Seurat they have started using Presto. Okay, this is a very C pops. It's written in C, very very fast way of comparing the differential gene expression analysis that they are by default. I think so they are using in Seurat 5. They are using Presto package to do that. Okay, and let's go to the alternate one. Find all markers. Here it does everything, but here we can do the filtering, like we can use minimum percentage cutoff 0.01 log to fold change, and which test to use, and whether you want only positive or not, and maximum cell that have expression. Those kind of parameters you can read and you can apply those filters as well. Okay, so let's do it try over here. So yep, so first here what we are doing, we are using FindMarkers function. Basically we are focusing on identity 2, that means second clusters. Okay, so if I just do that, what it will do, it will give me the cluster 2 markers. So now you can see what it indicates, like these are the genes, or these are the markers where p value is very low. And so generally we should not use p value, we should use adjusted p value. And what is adjusted p value? Because see, we have multiple genes, right? So we are doing so many tests. Okay, so that means because of doing so many tests, we are losing the statistical power of this p value. So that's why what we are doing, we are taking the p value and adjusting with the number of tests you perform. So let's say in this example, we are saying that we want let's say there are 100 genes we are considering for a DGE. So that means 100 times we are doing differential gene expression test. So p value will be we have to take the p value, and simplest is like you divide with 100. So number of tests you did, so that way your p value will be normalized or adjusted. But alternate one for any correction, different adjustment methods are there. But so that's what, when you are doing this multiple tests, right? So we need to consider this p value adjusted, not this p value, because this p value is a wrong one. The adjusted one is the because this is adjusted to the number of statistical tests that we are doing, and this accurately captures the statistical power or confidence of our each testing. Okay. Okay, so in this case, it says that this gene, the adjusted p value is good, less than 0.05, and the log to fold change is 6.9. That is log scale, that 6.9 very good. And it says that this gene particularly is expressed in 93% of cell in the cluster 1. It's like first this cluster, right? The 93% of cell that is expressed, whereas you see in this cluster it is expressed only in 0.04, means like it's not even 1%, right? So that's why you see the difference, and that's why this gene is the marker. So this gene is highly expressed in this cluster number 2, but rest of the group it did not express. Same you see here, this gene 85% of cells of the group 1 it is expressed, but in the group 2, the second clusters, expression was low. That's why you will see a high fold change. So more is the difference between this thing, the percentage expression as well, fold change. So these are the very different parameter we use, and using these three, again it's not a mean median kind of thing parametering. It's a rank based testing, and it does. So what is rank based testing? Just to give you some idea. So for example, if you take this gene, you in cluster 1 you rank the genes based on their expression value, and in the second group you also rank the gene based on the other value. Now if the CD791, what is the rank in the first group and what is the rank in the second group? So in this example, what will happen, the rank in the first group will be 1 because that's highly expressed. But let's say in the second group, because that gene is low expressed, the rank will be maybe somewhere, let's say 1000. So now you see the difference between this rank in the two group. So in the group 1 the rank is low, it's like 1. In the second group rank is very high. So that means the rank difference is good. So that indicates that okay, this gene is important. So if the rank in the both comparison group is same, right? So that means that gene is equally rank wise they're same. So that means those are not different. So any genes whose ranks are different between two group while arranging in terms of expression, those are the marker genes in the simplest way to understand. Okay, but Wilcoxon test is behind the scene. Okay, so let's move ahead. So this way we can identify 2 versus all. Again, you can also do like I want my first number group is cluster number 5 and my second group is not everything 0 and 3. So that way you are comparing 2 group. The group 1 is 5, the second group is 0 and 3. So that way you are comparing and calculating the FindMarkers function. Using that you are calculating. So these markers are cluster 5 specific. So you can see just an example, this gene in the cluster 5 the expression was 0.98, so 98% cells it is expressed. Again, the second group, that is in the cluster 0 and 3, they were less expressed. So that's why you see the difference, and fold change was around 8. So that's why this is very important. And adjusted p value is e to the power means 10 to the power minus 178. So it is highly significant. So these are definitely the marker for the 5th genes. Okay. So one by one it is doing very troublesome, right? So that's why we can use find all markers, and here you can specify that I want only the positive marker. So that means give me those genes or markers where that marker is only upregulated compared to the other group. So that way, so you can do that. So now it will see for every clusters it is now calculating the this thing. So we have now cluster 11 clusters, 0 is the first cluster, so it did that. So if you look at this PBMC, let's check the dimension. So you see that around this many differential gene expressed of all the clusters are there. Okay, so that's why what we can do, instead of doing, we can further do a filter, like first you group by cluster, the cluster, and then I want only those genes whose expression is greater than 1. So let's see how many we will get. Okay, so let's store this in X, or let's say IEXP something. Okay, so now check the dimension of IEXP. So this is now 7048. That means 7000 genes are there whose we see very high difference. But if I go with, let's say, 2, we will get some other value. Let's see. So now 3000 genes are there, and that to be, this includes all the clusters. Okay, got it. So now okay, so this is the way you can identify the important markers, okay, of each clusters. Better to do is, what we can do is, like, I'll just show you. You take this one, then group by cluster, and again this one, and you can say top 10. Okay, so this will give us for every cluster what is the top 10. So let's do, so not 10, let's say top 3. Okay, and just I'm doing view. Okay, what I did, you take the marker, group by clusters, and give a condition that only fold chain greater than 1, and just take 3 1 and just visualize this. So what it will do, for first 0 cluster, these 3 are the genes, marker genes. Second cluster, these are the genes. So only the top 3 genes for each clusters only I'm displaying here. Yeah. Okay, and further we can also do arrange by the fold to change, and we can apply some filter p value like that. Okay, so next, best is like to visualize. So there are different plots, as I mentioned, like the first one is the Violin plot. So if you know the genes, and then you can use Violin plot to see how the distribution. So for example, in this case Violin plot, you give the data and what all features you are want to see. Okay, so by default what it will do, okay, it will use the scale data, because already we have normalized and scale rate. It will use the scale data to show. Now it clearly says that this gene is expressed on the cluster number 2. Okay, whereas CD791 also expressed in the cluster number 2, because these are the cluster 2 markers. Okay, and you can also use the count, because so you can see slot equal to count, so it will display like this raw count, expression level. So that's why you see that the scale is very different here. This scale is 4 and 6 here, but in the raw count, when you use it, goes to 30 50, it will be that's why you can also use log equal to true. At least you will further scale down into a log scale, but it did not see much difference here. Only the this part got a little bit trimmed with the log scale. Okay, but yeah, so this is one way to visualize the Violin plot. Then second is the feature plot. Okay, so here the same thing, you just calculate the feature plot, and here what all things genes you want to see, visualize. Okay, so in this case you see, let me zoom it to explain you. Okay, so this is a UMAP plot. Okay, so UMAP clustering we have seen, right? So how does that look like? And but you can see that this MS4A1, this is specific to it, expressed in this cluster, and particularly this is the, I think, cluster number 2. Okay, and here GNLY, this part region or this cells only, this was expressed, and CD3, this cluster, you can see that. That's why you can see that this is not expressed in these 2 cluster, and that's why it's a marker of this group. And again that way, so that will, this way it helps us. Now this is a very interesting one. You see PPBP, this is just a very small cluster over here, it got expressed only that. Still very less number of cell, but that's a very useful signal. And these are actually, you can say that these are rare cell, because so many cells were there, right? And of which only, I think you can compare to that, this will be around 50-60 cells, but still the signal is very strong there. And those genes are pp bp is expressed only those genes, those cluster. Maybe you know about that, so you can say that, identify, oh, maybe few in your single cell data some clusters are belonging to this cell type, like that. Okay, so this clearly helps us to, on the UMAP projection, the clusters. Now you can also see how the expression moves around. Okay, so that is one thing. And alternatively, you can do a reads plot. Okay, so reads plot will give you same genes expression, like how the density plot kind of thing. So here you see the expression level here, and you see there is a peak over here, and you can clearly see on the second cluster that particular gene MSA41 has some higher expression value. And the same thing, it's like there is nothing fancy about it. So if you take this Violin plot, right, take this Violin plot and you cut it into half, you will get the reads plot. But it's a very nice way of representing. Okay, but I like this plot. For example, what you do, you do this one, like take the marker, you cluster them, good, put a condition, and top 10 you take, and just find the top 10 genes. So top 10 genes of each marker. Okay, so if I took top 10, right, you will see that these are the genes, top 10, right? Where are the names actually? So I think, okay, so we have the column called gene that contains the gene name. Okay, in the top 10, that. Okay, so now we can use this one to PBM. So we can do a heat map. And what features we are taking? We are taking the those genes. Okay, so what it will do, it will do the heat map of, again, for every cluster it took the top 10 genes. Okay, and then it tried to visualize the clusters as well. So now you can see that these genes particularly, they are expressed in 0, 1 and little bit 3 and 6 cluster. But this one, this particular gene set, they are expressed only on the cluster number 2. And these genes are expressed on cluster number 3 and cluster number 8, and this particular are expressed in 4 and 5. So like that. So that means these 4 and 5 will be very close in the UMAP plot. So like that, you can compare the UMAP projection as well as heat map and identify the markers, and strongly confidently say that whatever results you got from the differential gene analysis is also you are able to visualize that in the plots as well. So you need to combine the two information. You did the statistical testing to got those important genes. So now you take those genes and do this all visualization, and then see that actually what we are getting from the statistical analysis, actually you can see the UMAP and this heat map kind of plot or not. Okay, so that gives you extra confidence. And now once you are sure that, oh, okay, these genes are perfect one, now you go to the literature and do the next step, like identifying whether those genes have literature, what kind of cells those genes are expressed, whether B cell, T cell, like that, which is the next step what we will see. Okay, but the last I want to show is the dot plot. So basically you can specify these are the genes I want to see in a different way. So here these are the genes, these are the identity means the clusters, and the blue means higher expression, and whereas the gray is that they were not expressed at all. Okay, and size will be determined, how many cells, how many percentage of cells those are expressed. For example, this much size means almost 90% or 80% of cells in that 7th cluster, that is expressed like that. So dot plot gives 3 information. Not only you are getting the information gene cluster, but the color will tell how much is the expression, the size of the bubble, or that will tell is actually in how many percentage of cell those are expressed, sorry, not exposed. So in short, after PCA, you do the clustering, then do the visualization, then do compare, statistical analysis, and then identify the marker genes and visualize them. But next step is what? We need to annotate them. So now we got the marker genes. We know that, okay, these are the markers for cluster number 1. These are the markers for cluster 2, 3, 4, like that. So now we know the marker genes. So next thing what you need to do, you take those genes and search over internet, search over the literature, and try to find out whether anyone has annotated like, oh, hey, these genes are T cell, these genes are B cell, these genes are platelet cells, like that, So if it is known, then that will help us to do the cell type annotation, which we will see in the next video. So that's all about this. I think this is a very older slide. Yeah, I think that's all I had here. I hope this was useful. So thanks for watching. I will see you in the next video. Bye bye.